Thank you very much. It's a great pleasure and honor to be here. Particularly want to thank the organizer, ASM Tsinghua, and also Dr. Qijing Zhang, who's a good friend of mine, and it's truly wonderful time to be here in Beijing. So today I'm going to share with you some of the work we have been doing with Campylobacter. Since most of you do not work with Campylobacter, I want to give you a little bit of background information. This is a gram-negative organism. It's a sparky. It has a polar flagellum, and either on one end or both ends. It has a very small genome. It's only about 1.7 megabits, but it's very AT-rich, 70%. Campylobacter has many different species, more than 35 now identified, and it's macroaerophilic, means that it only need a little bit of oxygen, but not too much oxygen for optimal growth. Campy is very adaptive. It has a really broad host range. Pretty much every animal carry Campylobacter, domestic animals, wildlife, and human beings. But in many of these animal species, it's commensal, but in human host, it's pathogen. Now, the primary disease caused by Campylobacter in humans is enteric disease, diarrhea, watery and bloody diarrhea. But it also causes extraintestinal infections such as bacteremia, abortion, and other conditions. It also one of the primary factors associated with the Guillain-Barré syndrome, which is autoimmune disease. This pathogen has been increasingly resistant to clinical used antibiotics. Now, due to the importance of antimicrobial resistant Campylobacter, CDC listed this bug as a serious threat. That was the 2019 report. Now, as a zoonotic pathogen, Campylobacter can get into human by multiple routes, from contaminated meat, such as chicken meat, raw milk, contaminated water, petting zoo, and even companion animals, cats and dogs. In the US, the annual cases actually is more than mainly as estimated by CDC. Worldwide, more than 96 million cases of Campylobacter infection reported. Now, as a general step for Campylobacter to induce disease, I summarize here, it's transmitted through fecal oral route and first get into the GI tract, colonize in the intestinal tract, but some Campylobacter strains, not all of them, can translocate across the gut epithelium, get into blood circulation, and in pregnant animals, it may induce abortion. So that's the overall route of pathogenic steps of Campylobacter. So our lab has been studying Campy for many years, and in the past, I would say 8 or 10 years or so, we identified a hypervirulent clone of Campylobacter jejuni, which is ST8, MST type 8. This is a clone has been implicated in a number of human infections, outbreaks, sporadic infections. They're all foodborne diseases, and also has been reported to be responsible for bacteremia in the UK. And on the animal side, we found actually this clone has become the predominant cause of sheep abortion in animals. So responsible for more than 90% of sheep abortion is due to this special clone. So when we do the database search in the NCBI pathogen detection database using whole genome sequences, we found a number of matches, primarily from these human cases and also athletes from ruminants. This clone not only hypervirulent, but also is becoming multi-drug resistant to multiple antibiotics. So again, most of Campylobacter jejuni would colonize the gut as a local colonization, but some small number of strains would be able to invade and cause systemic infections. So ST8 is one of them. Now, when we first started to work on this clone, we tested the ability of ST8 in causing bacteremia using mouse model. You can tell this is IA3902 is isolate of ST8. You can tell it's, and in both the blood count and also liver tissues, ST8 has much higher titer compared to non-ST8 of Campylobacter jejuni. Now, in the abortion model with pregnant guinea pigs, ST8 is highly abortive efficient. These two isolate, all ST8 in isolates, and they induce nearly 100% of abortion in the inoculated guinea pigs. While these, these other Campylobacter jejuni strains, they only colonize the gut, did not induce any clinical abortion. So, the hallmark of this clone is systemic spread. When you analyze the tissues in infected animals, for example, these clone ST8, so we were able to find bacteria all over the place, from feces, bile, blood, uterus, and placenta, while these other, Campylobacter jejuni strains only can be isolated from the gut, the intestinal tract. So in the infected placenta, using immunohistostaining, we can detect lots of Campylobacter antigens. Now, the question we want to answer is what makes ST8 so virulent? So why this strain is so special compared to other, enteric strains? So, because since it induces systemic infection, the first thing we tested is whether a capsule is involved in the disease, in the pathogenesis. So indeed, when we knocked out capsule, this is a wild type, capsule mutant and a complement. And we tested the phenotype in the mouse model. You can tell definitely capsule in both the blood count and the liver counts in terms of bacterial number make a big difference. So capsules play a role in probably survival in the blood, and this is serum resistance again. The mutant strain definitely will not, a capsule mutant strain will not survive as well as the wild type and then complement strain. So, and in the guinea pig model for abortion, when we knock out capsule, the strain completely lost the ability to induce clinical abortion. So that demonstrates capsule is required for the pathogenesis, but it's not sufficient. The reason is, this ST8 produced capsule, but that's not the only strain. We have many other strains not causing clinical disease in pregnant animals, but also produce capsule. So this does not tell us the whole story. So we still wonder what is unique about ST8. Now, of course, everyone doing whole genome sequencing, right? So when we did that, we were very hopeful by analyze the genome sequencing, doing comparative genomics. Hopefully, we would be able to identify the specific virulent factors. But when we finished, we were quite disappointed. Actually, we didn't find anything particular, related to virulence. So the genome is highly synthetic to the non-abortive efficient strain, which is TRIP168. This does not cause any clinical disease in pregnant animals, and no apparent virulent factors, no pathogenic island, nothing really could tell us, in terms of pathogenesis. There is a plasmid, but the plasmid nothing to do with the virulence. We demonstrate that by curing the plasmid, kick out the plasmid, nothing, happens with virulence. So, and there are many different, of course, small changes, SNPs. But these small ones, you cannot really tell which ones specifically related to the virulence. So the bottom line is, but just by doing genomics, comparative genomics, that did not give us too much clues. So we figure out a strategy to, still to want to identify what are the specific gene exchanges associated with the hypervirulence. So that's the strategy we named directed genomic evolution. Essentially, it's a positive selection using the guinea pig model. So this is fairly straightforward strategy. We use the genomic DNA of ST8. We hypothesize it must carry the genetic materials related to virulence. Purified genomic DNA transform a non-abortifacient strain, which is NCTC 11168. So then we get these transformants. The transformants, just randomly happens, right? And we assume a small number of these transformants would gain the virulence associated genetic traits. So we put these transformants into guinea pigs. In the guinea pigs, as I said, majority of the non-abortifacient strains will stay in the gut, and only those transformants that has gained the virulence would be able to induce clinical abortion, right? So that's the positive selection. It's like a bottleneck effect. This step would filter out a majority of these transformants, nothing to do with virulence. Only allow these few that gained the virulent trait to be able to get into placenta and causes the clinical disease. So by collecting the aborted tissue, we were able to isolate Campylobacter from here, and we conducted whole genome sequencing, the output colonies compared with the donor and the recipient strains to figure out the specific changes in the chromosome. So first, we did whether the transformants would be able to induce clinical disease. Indeed, even a single transformation just makes the genomic DNA from the donor to the recipient. We convert 11168 from non-abortifacient to fully abortifacient. That's just a single transformation. For example, it's 11168 non-abortifacient, but once transformed with the genomic DNA from ST8 and fully virulence. These are the number of Campylobacter from tissue, from uterus, from placenta, and these are the abortion rate. You can tell basically the transformant is as virulent as the wild type ST8. Now we also did a quick macro restriction analysis of the transformants that caused clinical disease, and try to see whether there were large genomic rearrangement or genomic changes, but in this case apparently it's not. You can tell this is a recipient strain. All these transformants from aborted placenta tissue, they had identical fingerprints compared with the recipient strain, and totally different from a donor strain. That means these transformants, they gained the virulence, but did not have any significant changes on the chromosome. But when we did whole genome sequencing and compared the genomic analysis, we identified all these transformants, individual ones and pooled transformants from aborted placenta tissues. They all carry mutations in a single gene, which is PORE, encodes the major outer membrane protein in Campylobacter. So this protein actually is essential. Every strain has this PORE gene, but there are synthesis polymorphism. Each strain more or less is a little bit different. So ST8 actually were able to transfer all these SNPs into 11168 to make 11168 from non-virulent to fully virulent. So, and that's from compared genomics. We needed to verify indeed these transfer of mutations would be responsible for pathogenesis. Then we did allelic exchange. Basically, we swamped the single gene, PORE gene, from 11168 to 3902, which is ST8. Okay. Once we swamped the single PORE gene, we swamped the phenotype. So both ways, from virulent to non-virulent, from non-virulent to virulent. And these clinical abortion, the tissue distributions of Campylobacter. That's verify indeed the PORE mutations related to virulence. Now, when we further analyze these mutations, there are actually 19 of these amino acid changes on the surface of the major outer membrane protein. So most of them are in the external loops. These Morpheus outer membrane protein has these transmembrane segment and also externally exposed loops. So, then we further wanted to decide, are all these mutations required for virulence or just some of them? So we were able to make site-directed mutagenesis with different loops, loop 1, 2, 3, and 4. And I did, after we make these mutations, we did, animal challenge studies using guinea pig as a model. And to make the long story short, we found only loop 4 mutations, loop 4 here. This is, and there are 9 amino acid changes here. Only these amino acid changes would be required for virulence. None of these, others are not necessary. So that's, allowed us to nail down to the specific amino acid changes related to the virulence gain. Now, what is the significance of this finding? So definitely, previously, MOP is not known for virulence. People say it's porous, it forms a pore on the surface, allowing small molecules, nutrient, to pass through the outer membrane. And now we found a new function for this major outer membrane protein. So it's definitely related to pathogen-host interaction. And of course, we can utilize the SMPs for rapid detection of this hypervirulent clones. Design specific PCR or diagnostic test for detection of this pathogenic strain, and also potential candidate for virulence, for vaccines. If it is a virulent factor, would that be able to protect animals if we develop a vaccine using MOP? And lastly, it proves the utility of the DGE, which is a positive selection, a strategy to identify bacterial virulent factors. So we have, tons of genomics and comparative genomic data, but the bottleneck is for us is how to make sense of data. So I think this model can be very useful. So that's the identification of, MOP as a major virulent factor. And I want to also share with you about, the pathogen-host interaction in placenta, because that's the latest stage of the disease. Once Campylobacter survives in the blood, make it into a placenta, so what happens there? So we were able to take advantage of a sheep model. Sheep is a natural host for Campylobacter, and also once Campylobacter infects sheep, it not only induces clinical abortion, but the placenta carries tons of Campylobacter. So the CFU number actually can be 10 to the 8th or 10 to the 9th CFU program of placenta tissue. That would allow us to directly analyze the pathogen host response directly in situ without amplify the signals. So, and we did do RA-seq. That means we can analyze the transcriptomes from both the host side and also the pathogen side simultaneously right within placenta. So again, this is pretty tedious study. So just show a few slides. We actually identified a number of pathways are upregulated in the host side, but the most prominent one are these inflammatory and immune defense responses, and also antimicrobial defense mechanisms. Many of these genes in these pathways significantly upregulated. Now the downregulated, there's no pathways downregulated, but there are a few genes related to fetal tissue development are significantly downregulated. So that provides some clues for us. Maybe the reason for clinical abortion is related to upregulation, especially the massive stimulation of the pro-inflammatory response and also downregulation of fetal development. This is a chart shows how big of the change in terms of one pathway, which is cytokine receptor interaction pathway. All these red boxes are upregulated genes, so it's a massive stimulation of this pathway. And we were also able to verify the transcriptomic data using RA-scope, by doing in situ hybridization, TRR, IL-10. You can tell this is normal tissue, normal placenta tissue, and these the aborted placenta. So, and that's the host response. On the bacteria side, we were also interested what changed with Campylobacter in placenta, right? So when we did that, we found actually, again, many pathways changed. The most significant upregulators, iron acquisition, oxidative stress, heat shock. So maybe, this is not too surprising because once Campylobacter gets into the host, it has to compete with host for iron. So there are 5 pathways for iron acquisition and utilization in Campylobacter. All 5 went up. So, and the down-regulated ones related to energy production and cell cycle. So apparently, Campylobacter slowed down the metabolism and replication in order to survive. So that's the response in interactions in the placenta, and also want to share with you a little bit about because we are interested in the prevention and control of ST8 and also the evolutions and also epidemiology, especially molecular epidemiology of ST8. We wondered, what would be the commercially used vaccines out there? Would that work with this predominant clone? Because right now, more than 90% of the clinical abortions in sheep is related to this strain, right? So we there are 2 commercial vaccines in the US, A and B, and we also made experimental bacterium using ST8, which is homemade vaccine and sham control. So this is antibody response. All 3 vaccines induce good antibody response, no problem. They all immunogenic. But when you look at the protection, the commercial vaccine had a very little protection. So the commercial vaccine used on sheep farms had a very little effect on the predominant clone, while the bacterium remained with clone ST, which is fairly protective, it's 80% protection. So this means the vaccine used out there had a very protective rules against this clone. We also wondered, when did ST8 emerge in the US? Based on epidemiological observation, so it's fairly recent, but using the genomic data, we were able to obtain a number of historical isolates of ST8 from different years. The earliest one we were able to get is like in the late 80s, early 90s. Then, of course, we have a lot of recent isolate. By doing whole genome sequencing, we found actually ST8, which is also named clone SA, highly homogeneous, quite different from these non-ST isolates. And when we did a molecular analysis, it predicted its emergence around 1976. So it's about 50 or 60 years ago. This prediction actually is very, very close to epidemiological observation, because based on epidemiological observation, it's around early 80s. So it's a prediction and epidemiological observation all told us this is a recent clone. It's a very young clone. And we also using population genetics, trying to identify what has driven clone ST expansion. And again, PROA mutation is the only signal based on population genetics related to expansion of this clone. Again, and also TAT0, which is a tetracycline-resistant gene, the integration of TAT0 into the chromosome also directly correlated with ST expansion. So this means, probably use of antibiotics. Tetracycline is very commonly used for prevention and control sheep abortion out there, may have played a role actually in selecting of the emergence of this clone. So I want to just, summarize what I told you this morning. So definitely as zoonotic pathogen Campylobacter affect both animal health and human health, it's truly a challenge for One Health. Mutations in the major outer membrane protein actually affect the virulence of Campylobacter jejuni. It specifically convert Campylobacter from an enteric pathogen to a pathogen that can cause systemic infection as well as clinical abortion. So, and the reason we believe Campylobacter can induce abortions due to the stimulation, massive stimulation of pro-inflammatory response and also the downregulation of fetal tissue development. And also, we believe the expansion of emergence expansion of ST8 is a really recent event and has been driven probably by use of antibiotics as well as maybe the vaccine. The vaccine does not help at all. So the bottom line is if we can control ST8, this clone would benefit both animal health and human health. That would contribute directly to the One Health concept. With that, I want to stop and thank a few people, our people from the lab and the collaborators who have involved in this work, this multiple-year work. I take a little more than 20 minutes for me to share with you, but it took a number of years to finish. Thank you very much.
Thank you, Professor Zhang. We have time for a couple of questions. Yeah.
Hi.
Hi.
Very beautiful work, really.
Thank you.
I am interested to know, POR-A mutations, is it possible that it has been acquired by transformation from another bacterium, as have you done in your experimental work? Or did you see a sort of evolution of POR-A also in other STs related to ST8 along the time? So it was one event or an evolution in an horizontal direction?
That's a really good question. We believe it's the evolution. It's gradually accumulate these changes, but once it reached to this point, it's got fixed, during the evolution process. So we do not have any evidence to transfer from another bacterial pathogen by one step, horizontal gene transfer, or something is crossed, let's see, from Salmonella or E. coli to Campylobacter, or even from another Campylobacter species. So there's no evidence of that. So we believe it's the evolutionary process. Okay.
So great talk. So how do you think POR-A mutation makes bacteria more virulent?
I knew someone would ask that question. we do not have an answer yet. We have some hypotheses. We are still trying to figure out what these mutations do to the pathogenic steps. But we believe probably evolved in three possibilities: either translocation, the first step, from the gut to blood, or the second step, survival in blood, or the third step of inducing placental infection. Because porin, as a porin and also outer membrane protein, it could allow, for example, one possibility is allow the utilization of special nutrients, tropism to placenta. Campylobacter does not use glucose. That's the very unique. Only use amino acids, especially in the host. It prefers to use the amino acids. Pregnant animals has a lot of amino acids that might be very favorable for Campylobacter growth. So that's one possibility. And again, we cannot exclude other possibilities in terms of the function. But we don't know which exactly the step porin is involved. Maybe it's one step or three steps. That's something we're doing now. So.
Okay.
That's.
Do you have any case of this specific ST type to cause the abortion in humans in the United States?
In humans? No. We worked with CDC. CDC has a database with foodborne pathogens, but unfortunately, they don't have database with human clinical abortion. That's not very common. First, fortunately, it's very, very rare infectious abortion. So we don't have direct evidence. But in publications, Campylobacter jejuni as a species has been involved in human abortion, especially for pregnant women working with animals, like sheep farmers. There were some, sporadic cases of human abortion related to Campylobacter jejuni, but we don't have specific clue for this ST in the U.S.
Okay, great talk.
Good.
So I'm wondering, when you do RNA-seq, what kind of culture you compare in vivo and in vitro, right? But what kind of cultural condition you use for in vitro? Because that's a growth phase. What kind of growth phase you use? That's what determine.
Right. That's a wonderful question. So we, of course, we cannot. So in vitro, in vivo, really, it's totally different conditions. So we sequenced the Campylobacter, transcriptomics in placenta. We were debating, should we compare with stational phase or log phase, right? We did both, log phase, stational phase, and these changes consistent with both phases. And because another thing is Campylobacter does not have a typical stational phase response. The sigma factor for stational phase response does not exist in Campy. So it's a little bit different from Salmonella E. coli. So yeah, but a good question. No, this is the major purine. There's a minor purine. It's so this is only there are other purines, but there's a minor purine, but nobody has really sure it's a purine. So it's predicted to be purine, and expressed a very low number. But purine, this one is if you isolate outer membrane, you see a huge band. It's a major protein and a very distinct and expressed in high level in Campylobacter. Yeah. Thank you very much.